Culture media. The study of microbiology depends on the ability to grow and maintain microorganisms in the laboratory. A culture medium is a solid or liquid preparation used to grow, transport, and store microorganisms. Specialized media are essential in the isolation and identification of microorganisms, the testing of antibiotic sensitivities, water and food analysis, and other activities. Uses of culture medium. 1. To identify the cause of infection from clinical sample, so that proper treatment can be given. 2. To study the characteristics or properties of microorganisms. 3. To prepare biological products like vaccines, toxoids, antigens, etc. Types of culture media. Based on their consistency. Solid media, liquid media, semi-solid media. Based on constituents or ingredients. Simple media, complex media, synthetic or defined media, semi-defined media, special media. The special media are further divided into enriched media, enrichment media, selective media, indicator or differential media, sugar media and transport media. Based on oxygen requirements. Aerobic media and anaerobic media. Based on their consistency. A medium containing 2% agar as a solidifying agent is called a solid medium. Example, nutrient agar. In semi-solid media, the concentration of agar is 0.5% or less. Instead of agar, silica gel can also be used as certifying agent. A medium without a solidifying agent is called a liquid medium. Example, peptone water. Based on constituents or ingredients. Simple media. This media consists of only the basic ingredients for the growth of non-fastidious organisms. This media can be either solid, semi-solid or liquid media. Certain complex raw materials such as peptone, meat extract, and yeast extract are used, and the resulting media support the growth of wide variety of heterotrophic bacteria. Agar is included as a non-nutritive solidifying agent. Example, nutrient agar, nutrient broth. Synthetic or defined medium. These media are prepared from pure chemical substances, and the exact composition of the medium is known. Defined media are used widely in research, as it is often desirable to know what the experimental microorganism is metabolizing. Example, media for growing cyanobacteria and eukaryotic algae. Simple peptone water medium, with 1% peptone and 0.5% sodium chloride in water, may be considered as a semi-defined medium, since its composition is approximately known. Complex media. Media that contain some ingredients of unknown chemical composition are called complex media. Such media are very useful, as a single complex media may be sufficiently rich and complete to meet the nutritional requirements of many different microorganisms. Complex media contain undefined components like peptones, meat extracts, and yeast extract. Peptones are protein hydrolysates prepared by partial proteolytic digestion of meat, casein, soy meal, gelatin, and other protein sources. Beef extract and yeast extract are aqueous extracts of lean beef and brewer's yeast respectively. Most commonly used complex media are nutrient broth, tryptic soy broth, and McConkey agar. Special media. Media used for special purposes such as to select a particular organism, to differentiate between organisms, etc. are called special media. There are different types of special media. Enriched media. Blood and other special nutrients may be added to general-purpose media to encourage the growth of fastidious heterotrophs. These specially fortified media are called enriched media. Example: blood agar, chocolate agar. Enrichment media. Liquid media used to isolate pathogens from a mixed culture. Media is incorporated with inhibitory substances to suppress the unwanted organism. Example: selenite F broth for the isolation of Salmonella, Shigella. Alkaline peptone water for Vibrio cholerae. Selective media. This media favors the growth of particular microorganisms and do not enhance and may even inhibit other types of organisms that may be present. For example, a medium in which cellulose is the only carbon source will specifically select the growth of cellulose-utilizing organisms. Example: endo-agar, eosin methylene blue agar and McConkey agar. 3 media widely used in detection of E. coli and related bacteria in water, where the dyes in it suppress gram-positive bacterial growth. Differential media. These media distinguish between different groups of bacteria and even permit tentative identification of microorganisms based on their biological characteristics. Blood agar is both a differential medium and an enriched medium. It distinguishes between hemolytic and non-hemolytic bacteria. Hemolytic bacteria like Streptococcus will form a clear zone around their colonies due to RBC destruction. McConkey agar is both a differential and selective medium. It differentiates lactose fermenters and non-lactose fermenters. It contains the neutral red dye. Lactose fermenters appear pink to red and others colorless. Indicator media. This media contains an indicator which changes color when a bacterium grows in it. Example: Wilson-Blair medium. Salmonella typhi reduces sulfide present in the medium to sulfide in the presence of glucose, and the colonies of Salmonella typhi have a black metallic sheen. Transport medium. In the case of delicate organisms like gonococci, which may not survive the time taken for transporting the specimen to the laboratory or may be overgrown by known pathogens, special media are devised for transporting the specimen. These are termed transport media. Example: Stod's medium. It is a non-nutrient soft agar gel containing a reducing agent to prevent oxidation. Sugar media. The usual sugar media consist of 1% sugar in peptone water along with an appropriate indicator. A small tube, Durham's tube, is kept inverted in the sugar tube to detect gas production. Anaerobic media. This media is used to grow anaerobic organisms. Example: Robertson's cooked meat medium. It is a liquid medium with finely divided meat pieces which act as reducing agents.